antibody, let me introduce you what is antibody. An antibody is a Y-cell immunoglobulin protein of our immune system. It recognizes the foreign objects such as bacteria and viruses. Antibody hooks to a unique part of the foreign target called epitope by its unique structure on the tip of Y called paratope. Antibody, paratope, antigen, epitope. Binding is a lock and key precision mechanism that results in neutralization of the foreign objects. Okay, so how do we prepare the antibody? You can make antibody in any animals. For example, here, a goat, sheep, a rabbit, mouse, anywhere. What you do, you take the protein or antigen of your interest where you want to make antibody, put it in any animal, inject it to them, and after a few weeks, you draw the serum, and that is your antibody. Now, this is called polyclonal antibody because it recognizes many of the sites or epitopes in the protein or antigen. And the limitation of this is you have a limited amount of antibody in this case because once serum is finished, antibody is finished. But if you want a very specific monoclonal antibody and if you want an unlimited amount of stock, then you can make a monoclonal antibody in mouse. So what you do here, you inject your antigen or protein to mouse and take this antibody producing plasma cells and fuse it with long-lived plasma cells and make a hybridoma. So once you have a hybridoma, you can put it in a culture and just grow them and take the anti-serum, and that is a monoclonal antibody, unlimited amount, unlimited supply. And if you are lucky, that is it. Now, just to remind that this is a cell, this is a nucleus, and on the plasma membrane there are proteins, and some are static, and others are unstatic. Unstatic means they may go in, like here, they may go in, and that is called internalization. So you can imagine if this protein goes in, and if we tag this protein or antigen or epitope to our antibody, this antibody will also go inside the cell. And if you tag some fluorochrome to this antibody or some drug, then this whole thing will go inside the cell. And that will be very useful, this internalizing antibody will be very useful for potential nanocarriers for delivery, imaging, therapy, and so on. Now, I will introduce the antibody against the antigen or protein called motley. You must have heard about motley in the morning session, plenary lecture. Just to remind you that motley was cloned in 1993 in our hands in our laboratory. The other name of motley, this protein is MT-HSP70, GRP75, PBB74, and it is a multi-talent dynamic protein. It helps in mitochondrial biogenesis, chaperoning, intracellular trafficking. Trafficking means the antibody comes to the plasma membrane and collects the protein which wants to travel to nucleus, it brings to nucleus and leaves it. So that is called trafficking. And it also has a role in pro-proliferation. Pro-proliferation means in cancer. It has a role in promoting the cancer. So it also helps in stress management and so on. And this motley protein has many binding partners. And the important is where it is localized inside the cell. That is important. But this motley protein is found in mitochondria, endoplasmic reticulum, plasma membrane, and also in the nucleus. Now, if you look at the cell, normal mouse cells, this is a cell, and this is a nucleus, around here black one, and this is a cytoplasm, and this is a cancer cell, mouse cancer cells. So if we study the motley protein, we find that motley in normal cells is all around the cytoplasm. But in cancer cells, it is around the nucleus. So you can see from this cartoon, motley is here all around the cells, in the normal cells, and in cancer cells, it is around the nucleus, also called perinuclear. Now, we have raised the antibody against this protein, and in human cells, if you look at by Western blotting, Western blotting is a method where you load your protein in an electrophoresis and then transfer it to a membrane and probe it with an antibody. So by Western blotting, what we find that this antibody picks up one band here in normal human cells and cancer cells like bone cancer, U2OS and SRS2 cells. And if you look at the staining again here in human cells, we get the same pattern of staining what I showed you in the mouse cells. Like in normal cells, it is in the cytoplasm all over. In cancer cells, it is peripheral. So this is the distinction. Just with the antibody, you can distinguish this is a normal cell and this is a cancer cell. Now, since mottling is a heat shock protein, we have to check whether this antibody is very specific to the particular protein or it is reacting to other proteins. So to do that, we heat shock the cells, and by single electrophoresis, so one-dimensional, we find that it picks up one band, and in two-dimensional SASGS polyacrylamide gel electrophoresis, when heat shock is given, there are so many proteins are induced, but this antibody picks up only one spot here. So which means this mottling antibody is very specific. So once we have antibody very specific in hand, then we go for immunostaining. So all of my talk now is about immunostaining. So I would like to introduce you what is immunostaining. What we do, we culture cells in a petri dish or this slide or cover slip here and fix them after some population of when cells are grown. We fix them there. There are different types of fixative, standard method. Then we incubate them with the first antibody. In this case, we are using anti-mottling antibody. So after incubation for an hour, we do the washing to remove all the unspecific binding and then go for the second antibody, incubation. So second antibody should be specific to the first antibody. If you are using your rabbit first antibody, it should be rabbit and second antibody. And then this second antibody is tagged with the fluorochromes like FITC, Texas Red, so on. And then once this incubation is done, we go for microscopy. So when we go for microscopy, either we can observe one protein at a time, or we can observe two or multiple. So if we are looking at multiple, if one protein we tag with one fluorochrome like green, other with red, then we can see like these colorful visual. Now this is one example to show that this is a cell, this is a nucleus here with almost here, and this is a one protein tubulin. So this is a multicolor. In vitro, in cell staining, this is in a tissue staining. So this is how it looks like if you do multiple staining. Okay. Now, since I will be talking about cell internalizing antibody, that means antibody going inside the cells without any help. So in this case, we have modified the protocol. What we have done here, so we add antibody here directly, and we skip this part. So this time, all incubation is skipped. So we go from here to washing, and then since first antibody is here, we directly go to incubate the second antibody and do the same process. So what we see that anti-mottling antibody, if we add to the cell, fix them, and stain with the second antibody, then they are stained. That means the first antibody has gone inside. So this is how it looks like. This is a bone cancer cells, and this is a breast cancer cells. These are cells. These black dots here is small round nucleus. So they are perinuclear. Here also perinuclear. As I said before, cancer cells have a perinuclear protein distribution.